Okay, hi everyone. Okay, so hopefully you've already started these notes, but if not, here you go. Okay, so these are going to go on page 73, 70 through 75 under the warm-up because today's warm-up went on page 75. So we're going to start with the patterns of inheritance. What are some exceptions to Mendel's principles? Mostly, we found out later on as we kept on studying um, chromosomes and how inheritance and the study of genetics, as that continued and got more intricate over time and we got better technology, the examples of it became clear how other genes that did not fit Mendel's original studies, how those get passed on, and that is what we're going to cover in this section. So you don't have to write down the slide except for the part that defines classical dominance. So we've studied classical dominance already, and classical dominance states that some alleles are dominant and they hide the recessive allele. In other words, the capital letter the one allele, the one we use an uppercase for, um, hides or overshadows overlaps the lowercase or in the dominant allele. For example, here we have Minimi. If he was like if you had the rock standing in front of him, you wouldn't be able to see the recessive Minimi. So that we see that case happening in dominant and recessive, or in eye color, we see brown. Generally, when someone gets at least one brown dominant gene, they will have the brown gene phenotype. So it'll display like some kind of brown eyes. In order to display the blue eyes, you would have to have the no dominant around at all. And in this chart right here, you can see that in classical dominance, when you have the homozygous dominant or the big B, you wind up with brown eyes. If you have the heterozygous big B, little b, you get the brown eyes also. And then you get the homozygous recessive, that's the only case you would get blue eyes. So in classical dominance, there are three genotypes and two phenotypes, and that's what we see, what we studied so far. You do not need to take notes on this part, but we do not see classical dominance all the time. We see all kinds of dogs and cats that have multiple color spots, different colors all over the place. Uh, we see different situations in nature that don't fit classical dominance. And incomplete dominance is an example. And yes, you should write everything down from this slide. Incomplete dominance is one allele does not completely dominate the other. In other words, the heterozygous genotype, the RW, is a mixed, and you can see here in snapdragons plants, They produce, if you get one parent that produces a homozygous dominant or big R red flower, and you were to breed it with a white flower, in this case would be WW. Notice we use all capital letters here. We wind up with their kids all being pink. And so this is a mix. Okay. Incomplete dominance, they mix in. They're all mixed in. So incomplete dominance mixed in. That's what you can use that to remember that the incomplete dominance, it's mixed in, incomplete, it's mixed in. You can remember, that's a way to remember it phonetically, because this one blends together. Okay, in this slide you write the only these two parts. Blending of traits, red and white make pink, and that's basically what's describing what's happening in the picture. And you do not write where it says pink, red does not completely dominate the white, or the white does not completely dominate the red. You would not add that to your notes. You would only write the red and white traits are incompletely dominant and blend together to make a pink. So they're mixed in, and that's the explanation for incomplete dominance. The offspring are mixed. So, genotypes and phenotypes. In this case, you see a clear difference from classical dominance. Here we've got the capital R homozygous dominant still gives you red. The capital W still gives you white, but when you combine them, you wind up with they're mixed in. So you've got pink. So in this case, incomplete dominance, there are three genotypes and three phenotypes, not three and two like we saw in classical dominance. You should practice these just to make sure you understand what incomplete dominance is like, and how to make um, complete these problems. You will also have these in your homework, so hopefully that would help. These will help you. Okay, so now we have another case that does not fit classical dominance. This is called codominance. Both traits or alleles share the dominance. Both traits are expressed, and here we've got a little plant that has leaves that have little moons on them, and a little plant that has leaves that have little squares on them, and if you blend them together, you wind up with little baby plants that have both moons and squares. And that expresses what codominance is. And anytime you see stripes or anytime you see spots on an organism, that is an example of codominance. Okay, so whenever you think of codominance, think of co-workers or co-champions that you share. So there is no true dominance. Basically, you get co-dominant traits are both dominant. So you get no recessive. There's just dominance. So both traits are present. So we're going to go through an example that you're going to have to do. Your homework is based off of right here. So blood is a blood type is a codominant trait. So this is an example of codominance found in us. There are four types of blood that our people have. They have type A, type B, type AB, and type O. And they A and B are codominant. That's why you have a type AB. And in this case, you also have a recessive, so type O, and that's what the classic recessive is. So not only do we have like a it's like codominance with classical dominance combined here. So in this slide, you please draw the pictures if you haven't drawn them already. So type A antigens. What's an antigen? An antigen is basically a name we give a surface protein. So on the surface of some blood cells, you get these little antigens, like the little green ones on A that look like little kind of boxy. You can draw them as little squares on your drawing, and that's the kind of proteins they have on the outside. If you have type B blood, your antigens don't look like the type A antigens. They look like type B antigens, which are little triangles. If you get type AB, if you have the genotype AB, you produce both little boxes, type A antigens, and little triangles, type B antigens. And if you happen to have type O, you don't produce any antigens, and that's the recessive. If you get two recessives, you don't produce any antigens at all. So what this means is that if you have type A antigens, your body has white blood cells that will attack anything that doesn't have type A. Or those type of antigens. So if someone tries to put type B into your circulatory system, and I mean don't mean like a little bit, like you're going to cut yourself and you touch somebody else's cut and oh you get some B type of blood. No, somebody like you lost a lot of blood and somebody tries to transfuse a lot of blood. That's not type A into you. Type B, what it does is that your white blood cells will attack those cells, and basically if there's enough there, it can actually you'll spike a fever and die within a couple of hours. Same thing for B. If you're type B and someone injects a lot of type A into your body, will fight it to the point if there's enough there, it'll kill you. So it's really important that they give you the same blood that you're supposed to have. If you type AB, if because your body knows what antigen A looks like and your body knows what antigen B looks like, or feels like, your body will not attack anything that has antigens A or anything that has antigen B. So your body won't be triggered to attack for A or B because you've got them both. Now, in the case of O, if you're type O and someone gives you A, it's bad. If someone gives you B, it's bad. A, B is bad. So, and if you're someone that are type O, your body cells don't have, your red blood cells don't have any antigens. That's what your body knows. So, any kind of antigen will be catastrophic to you. So each antigen or surface protein is unique and they do not mix. This is a reason why only certain blood types can be donated or accepted by other blood types. And I just talked to you guys about that. So if you have type A blood, then your body can only accept A antigens. So when you need blood, you can only accept from other type A or type O because there is no antigens on O. And that's really important about O. O, because it's a recessive and because it does not have any antigens, basically there's nothing for the body to attack if you to attack. So if O gets put into anything else, there's nothing for them to attack. Okay, so make sure you make this chart and then fill it in. Okay, so blood type A, B, and O. So here it's an acceptance chart, donating chart. So type A can get from A and O and donate to A and AB. Type B can get from B and O. Type AB can get from type AB, and O. So basically from all of them. Type O can get from type O only. Um, to donate, type A can only donate to A and AB. Type B can only donate to B and AB. Type AB can only donate to AB, and type O can donate to AB, and O. So make sure you write down this chart in your notes. Okay, and finally, blood types are also follow the rules of codominance, so you should have this in your notes. Type A, so phenotype and genotype. The phenotype A, you would have to be either homozygous dominant for A or heterozygous for A. Type B, you have either homozygous B or B basically O. If you are type AB, you have to have A and B. If you have type O, you have to have OO. Okay, so those are the genotypes and the phenotypes associated with blood types. There is a case of codominance here, and you have a case of clear dominance and recessive because A and B are codominant and the O is the recessive.